Hello students, welcome to today's lesson on HIV/AIDS. In this lesson, we are going to study some features, structures, and reproduction mechanisms of HIV. What is HIV? HIV is an abbreviation that stands to H human, I immunodeficiency, V virus. As the name tells us, HIV virus is a retrovirus that damages human immune system. Since the host cells to HIV is immune cells, it results in deficiency of immune cells to the infected person. This is the reason that the name of the virus is HIV, that means human immunodeficiency virus. Since it damages the immune system, it makes the infected person to face deficiency of immune cells. This is what the name tells us, human immunodeficiency virus. This virus grouped under retrovirus. Being retrovirus in a sense means it has RNA as a genetic material that can be reversely transcribed to DNA once it gets injected into the host cytoplasm through the action of reverse transcriptase enzymes. Here, reverse transcriptase enzymes are responsible to convert viral RNA to viral DNA. These are an exceptional features of retroviruses. Since it converts RNA to DNA means reversely transcribe RNA to DNA against the normal natural process or central dogmas. We know that the normal genetic information flow in all living organisms is transcribing DNA to RNA. But in case of retroviruses, they reversely transcribe RNA to DNA. When we see the host organism to HIV, the host to HIV is human being or Homo sapiens. After this HIV gets inside into the host organism, human being, it primarily targets CD4 cells of human being and most particularly T helper cells. As we said, HIV infects and kills CD4 cells until very few are left. It most particularly impair or damage T helper cells. Without these very important or crucial T helper cells, the body cannot mount or control a defense mechanism against the invading bacteria or viruses. This is the reason that AIDS patients die of infection that a healthy person could fight off or cope up. What is AIDS? AIDS stands to Acquired Immune Deficiency Syndrome. This disease is caused by HIV. As the name Acquired Immune Deficiency Syndrome tells us, a person that has AIDS shows syndromes or signs of deficiency of immune cells, while a person that is AIDS patient shows syndrome or signs of deficiency of immune cells, because HIV causes a severe damage to the immune system and it limits the body's ability to fight infection due to markedly reduction of T helper cells through the action of HIV. In this case, patients have a very weak immune system or defense mechanism against any foreign intruders. Due to this reason, patients predisposed or exposed to multiple opportunistic infection that leads the patient to death. These opportunistic infections that lead the patient to die are pneumonia, cancers such as Kaposi sarcoma, and central nervous system lymphoma. When we see structures or anatomies of HIV, there is central core, which is made up of two identical strands of RNA and reverse transcriptase and integrase enzymes. The second one is capsid or capsule proteins that's made from glycoprotein 24, which is responsible to enclose or wraps around the central core or genetic material. This capsule protein or capsids must be gets enclosed by matrix protein that's made from glycoprotein cell. Okay, the next one is the outer coat that encloses the matrix protein. This outer coat of the virus is known as viral envelope, and this viral envelope is composed of two layers of fatty molecules called lipids. These viral envelopes are taken from the membrane of a human cell when a newly formed virus particle buds from the host cell. Due to this reason, the envelope is fatty coat and has 72 little spikes that protrude out from the envelope. Spikes are complementary proteins on the CD4 proteins of T helper cells. The spikes on the surface of HIV are made from 3 glycoprotein 120 molecule that attached to another molecule called GP41. The shape of these spikes allows the virus to bind with CD4 receptors on the T helper cells or the host cells. To remind you again, the spikes on the surface of HIV are made from 3 glycoprotein 120 plus glycoprotein 41. Glycoprotein 120 is sticking out of HIV that allows HIV to bind with CD4 receptors of T helper cells or CD4 lymphocytes, macrophages, and some natural killer cells. The reproductive cycle of HIV. HIV replicates to cause disease when it gets inside into the host cell. The first stage to replicate itself is attachment. HIV particle first needs to attach itself on the surfaces of the host cell, CD4 receptors. Then glycoprotein 120 gets bind with CD4 receptors on the host cells. That means glycoprotein 120 is sticking out of HIV virus particle connects with CD4 receptors sticking out of CD4 cells like an egg fitting into the egg cup. Once the virus has attached to the host cell, it can go on to the next stage and merge with the host cell. After it merges to the host cell, the virus fuses and penetrates to inject its genetic material RNA with other enzymes like reverse transcriptase enzyme and integrase enzyme. Once inside the host cell, HIV particle sheds its protective coat, capsid. This leaves viral RNA floating in the cytoplasm along with reverse transcriptase enzymes. Therefore, to use its genetic material RNA for replication, HIV protective coating surrounding RNA must be dissolved. Once the protective coats, capsule protein P24 and matrix protein P17 gets dissolved, reverse transcriptase enzyme uses building blocks from the T helper cells to synthesize a double strand of DNA complementary to the virus RNA. During this stage, reverse transcriptase often making mistakes and introducing new mutations. That means viral RNA gets reversely transcribed to viral DNA through the action of reverse transcriptase enzymes. Once viral DNA gets synthesized or reversely transcribed, it must be transported across the nucleus and integrated into the host DNA through the action of integrase enzyme. Once this viral DNA gets integrated with the host DNA, the viral DNA hides itself into the host DNA and gets replicated when the host's DNA gets replicated for a certain periods of time until some triggering environmental conditions gets appear. When some triggering environmental condition gets appear, this viral DNA gets transcribed to viral RNA and controls viral protein synthesis in the cytoplasm. That means the integrated viral DNA controls the synthesis of viral RNA, and hence viral RNA controls synthesis of viral proteins. Now, all the required materials, that is, viral protein and viral RNA, are all get synthesized, and these materials must be separated or cleaved and assembled into new HIV particles. These new HIV particles gets approached on the inside walls of host cell membrane. Separation or cleavage of the required proteins, the required sizes of protein to produce viral particle is simply gets performed by protease enzymes. That means protease enzymes here chop up or break off viral RNA and viral proteins to make viral particles, HIV viral particles. Then lastly, these HIV particles leave the host cell through budding. That means the newly formed HIV particle pinches off the host cytoplasm and live by being enclosed with CD4 cell membrane as its envelope. Okay, the next one is the latency period. Even though HIV leaves the host cell through budding or pinches off by chronic ongoing release cycle without damaging or killing the host cell, some HIV proteins remain on the surface of the infected CD4 cells, and then these HIV proteins can be recognized by some immune system cells. These cells gets destroyed by natural killer cells. Even though HIV do not burst the host cell, our immune cells recognize this host cell as foreign matter and then destroy this host cell. This is the reason why our immune cells gets reduced in number when HIV grows in our body. The latency period is the period when the body keeps replacing CD4 lymphocytes as fast as they are destroyed by HIV. This latency period can last for many years. Characteristically, HIV infection can progress for 10 to 20 years before clinical syndromes or AIDS gets appear. The cycle of infection, reproduction, and destruction of infected cells repeats itself for as long as the body can keep replacing CD4 lymphocytes. Eventually, the body will not be able to replace these cells, and the number of free viruses in the blood will increase dramatically. HIV may infect other areas of the body, including the brain. Because of the drastic reduction in the number of T helper cells, the immune function is severely reduced, and many opportunistic infections may occur, including pneumonia and tuberculosis, and sometimes or rarely cancers like Kaposi sarcoma. So the period when the body keeps replacing CD4 lymphocytes as fast as they are destroyed is called latency period and can last for many years. Thanks for watching. If you like this video and want to get the next video, subscribe, comment and share this video.